Welcome to EPG Padasala. I am Dr. Gayatri Devi from the Department of Biochemistry, Biotechnology and Bioinformatics. I am working as Associate Professor in Avinash Lingam Institute for Home Science and Higher Education for Women. Today in this class we are seeing about synapse and conduction of nerve impulse. To introduce, synapses are defined as the neurons communicate with one another at junctions. At a synapse, one neuron sends a message to a target neuron of another cell. Neuron from which the axon arises is called the presynaptic neuron, and the neuron on which the axon ends is called the postsynaptic neuron. Synapse is only a physiological continuity between two nerve cells and not an anatomical continuation. Most of the synapses are chemical, which communicate using chemical messengers, whereas other synapses are electrical, and in these ions flow directly from the presynaptic cell to the postsynaptic cell through gap junctions. This module is designed to give about synapse and conduction of nerve impulses. Synapses are the junctions where signals are passed by one neuron to another neuron, muscle cells or gland cells. Most of the nerve-to-nerve signaling and all well-known nerve-to-nerve or nerve-to-muscle and nerve-to-gland signaling depends on chemical synapses at which the presynaptic neuron releases neurotransmitter which acts on the postsynaptic target cell and make it more or less likely to fire an action potential. After completing this module, you will be able to know the role of synapse in transmitting nerve impulse, to learn the classification of synapse and its functions, to understand the mechanism of transmission of impulse by synapse. First, let us see about the classification of synapse. It is classified by two methods as anatomical classification and functional classification. What is anatomical classification? The synapse is formed by an axon of one neuron ending on the cell body dendrite with axon of the next neuron. The synapse is classified into three types depending upon the ending of axon. They are axonic synapse, in which the axon of one neuron terminates on axon of another neuron. Axodendritic synapse, in which the axon of one neuron terminates on dendrite of another neuron. Third one is axosomatic synapse, in which axon of one neuron ends on soma of another neuron. The following figure clearly explains these anatomical synapses. Coming to functional classification, it is the synapse is on the basis of nature of impulse transmission. According to this, synapse is classified into two categories as electrical synapse and chemical synapse. Electrical synapse is a synapse in which the specialized physiological continuity between the presynaptic and the postsynaptic neurons is provided by gap junctions between the membranes of two communicating neurons. There is a direct exchange of ions between the two neurons through the gap junction. Owing to this, the action potential reaching the terminal portion of presynaptic neuron directly enters the postsynaptic neuron. This type of impulse transmission occurs in some tissues like the cardiac muscle fibers, smooth muscle fibers of intestine and the epithelial cells of lens in the eye. Figure 2 explains the electrical synapse and the chemical synapse. Chemical synapse is a special junction through which the neurons signal to each other and to non-neuronal cells like muscles and glands, such as transmission of nerve impulse between a nerve fiber and a muscle fiber or between two nerve fibers, through which the signals are transmitted by the release of chemical transmitter. In the chemical synapse, there is no continuity between the two neurons because of the presence of a space called synaptic cleft between the two neurons. The action potential reaching the presynaptic terminal causes the release of neurotransmitter substance from the vesicles of this terminal. Neurotransmitter reaches the postsynaptic neuron through synaptic cleft and causes the production of potential change. Coming to the functional anatomy of chemical synapse, the neuron from which the axon arises is called the presynaptic neuron, and the neuron on which the axon ends is called postsynaptic neuron. Axon of the presynaptic neuron divides into many small branches before forming the synapse, and these branches are known as presynaptic axon terminals. Now let us see about the types of axon terminals. Terminal knob. Some of the terminal knobs are enlarged slightly like knobs are called terminal knobs, and they are concerned with the excitatory function of the synapse. Terminal coils or free endings. The other terminals are wavy or coiled with a free ending without the knob. These terminals are concerned within the inhibitory function. The structures of axon terminal and presynaptic membrane. The presynaptic axon terminal has a definite intact membrane known as presynaptic membrane. Axon terminal has two important structures. Mitochondria. It is one of the nerve cell organelle which is responsible for the supply of energy in the form of ATP. Here also, the mitochondria form the powerhouse of the nerve cell. For example, in the neuronal membrane, the special protein uses the energy released by the breakdown of ATP into ADP to establish concentration differences between the inside and the outside of the neuron by pumping certain substances across the membrane. It also helps in the synthesis of neurotransmitter substances. Second one is synaptic vesicles. They are also called as neurotransmitter vesicles. In a neuron, various neurotransmitters are stored and released at the synapse. The release of neurotransmitter is regulated by voltage dependent calcium channel. The vesicles are essential for propagating nerve impulses between neurons and are constantly recreated by the cell. The axon terminal or button is the area where the axon that holds as a group of vesicles. Up to 130 vesicles can be released per button over a 10-minute period of stimulation at 0.2 Hz. In the human brain region, B1 synaptic vesicles have an average diameter of 39.5 nanometers with a standard deviation of 5.1 nanometers. The two classes of obligatory components of synaptic vesicles are called transport proteins, which are involved in neurotransmitter uptake, and trafficking proteins that participate in synaptic vesicle exocytosis, endocytosis, and recycling. The transport proteins are composed of proton pumps that generate electrochemical gradients, which allow for neurotransmitter uptake, and neurotransmitter transporters that regulate the actual uptake of neurotransmitters. VATP is the enzyme which breaks down the ATP for energy to create necessary proton gradient. Vesicular transporters move neurotransmitters from the cell's cytoplasm into the synaptic vesicles. Vesicular glutamate transporters, for example, sequester glutamate into vesicles by this process. Trafficking proteins are more complex. They include intrinsic membrane proteins, peripherally bound proteins, and the proteins such as SNAPs. These proteins do not share a characteristic that would make them identifiable as a synaptic vesicle proteins, and little is known about how these proteins are specifically deposited in synaptic vesicles. Many, but not the entire known synaptic vesicle proteins, interact with non-vesicular proteins and are linked into specific functions. The synaptic cleft and postsynaptic membrane. The membrane of the postsynaptic neuron is called postsynaptic membrane, and it contains some receptor proteins. The small space in between the presynaptic membrane and the postsynaptic membrane is called synaptic cleft. The basal lamina of this cleft contains choline esterase which destroys acetylcholine. Now let's see about the functions of synapse. The main function of the synapse is to transmit the action potential from one neuron to another. Although some of the synapses inhibit these impulses, they are not transmitted to the postsynaptic neuron. On the basis of functions, synapses are divided into two types, excitatory and inhibitory. Excitatory synapse which transmit the impulses, that is excitatory function, and inhibitory synapse which inhibit the transmission of impulse, that is inhibitory function. This excitatory function or postsynaptic potential, that is during the process of synaptic transmission, non-propagated electrical potential get developed and is called as excitatory postsynaptic potential epsp. The voltage gated calcium channels at the presynaptic membrane are opened once the action potential reaches the presynaptic axon terminal. Now the calcium ions enter the axon terminal from extracellular fluid. Calcium ions cause the release of neurotransmitter substance by means of exocytosis from the vesicles. Neurotransmitter, which is excitatory in function, passes through presynaptic membrane and synaptic cleft and reaches the postsynaptic membrane. Now, the neurotransmitter binds with receptor protein present in postsynaptic membrane to form neurotransmitter receptor complex. The neurotransmitter receptor complex causes production of a non-propagated epsp. Common excitatory neurotransmitter in a synapse is acetylcholine. The neurotransmitter receptor complex causes opening of ligand-gated sodium channels so that the sodium ions from the extracellular fluid enter the cell body of postsynaptic neuron. As the sodium ions are positively charged, resting membrane potential inside the cell body is altered and mild depolarization develops. This type of mild depolarization is also called as epsp. From here, the action potential spreads to other segment of the axon. In neuroscience, an excitatory postsynaptic potential epsp is a postsynaptic potential that makes the postsynaptic neuron more likely to fire an action potential. This temporary depolarization of postsynaptic membrane potential caused by the flow of positively charged ions into the postsynaptic cell is a result of opening ligand-gated ion channels. These are the opposite of inhibitory postsynaptic potentials, that is IPSPs, which usually result from the flow of negative ions into the cell or positive ions out of the cell. epsps can also result from the decrease in outgoing positive charges, while IPSPs are sometimes caused by an increase in positive charge outflow. The flow of ions that causes an epsp is an excitatory postsynaptic current. epsps, like IPSPs, are graded, that is, they have an additive effect. When multiple epsps occur on a single patch of postsynaptic membrane, their combined effect is the sum of the individual epsps. Larger epsps result in greater membrane depolarization and thus increase the likelihood that the postsynaptic cell reaches the threshold for firing an action potential. Now we are moving on to excitatory molecules. The neurotransmitters most often associated with epsps is the amino acid glutamate and is the main excitatory neurotransmitter in the central nervous system of vertebrates. Its ubiquity at excitatory synapse has led to it being called the excitatory neurotransmitter. In some invertebrates, glutamate is the main excitatory transmitter at the neuromuscular junction. In the neuromuscular junction of vertebrates, EPP, that is the end plate potentials, are mediated by the neurotransmitter acetylcholine, which along with the glutamate is one of the primary transmitters in the central nervous system of invertebrates. At the same time, GABA, that is gamma aminobutyric acid, is the most common neurotransmitter associated with iPSPs in the brain. However, classifying neurotransmitters as such is technically incorrect, as there are several other synaptic factors that help to determine the neurotransmitter's excitatory or inhibitory effects. The properties of EPSP. It is confined only to the synapse. It is a graded potential. It is similar to receptor potential and end plate potential. This EPSP has two properties. It is non-propagated and it does not obey all or none law. On seeing the significance of EPSP, it is not transmitted into the axon of the postsynaptic neuron. However, it causes development of the action potential in the axon. When EPSP is strong enough, it causes the opening of voltage-gated sodium channels in the initial segment of axon. Now, due to the entrance of sodium ions, the depolarization occurs in the initial segment of axon and thus the action potential develops. From here, the action potential spreads to other segment of the axon. The following figure shows the sequence of events during synaptic transmission. We are now moving to inhibitory function. Inhibition of synaptic transmission is classified into 5 types. Postsynaptic or direct inhibition, presynaptic or indirect inhibition, negative feedback or Renshaw cell inhibition, and feed-forward inhibition and reciprocal inhibition. First, let us see about the postsynaptic or direct inhibition. It is a type of synaptic inhibition that occurs due to the release of an inhibitory neurotransmitter instead of an excitatory neurotransmitter substance from the presynaptic terminal. It is also called direct inhibition. Inhibitory transmitters are gamma aminobutyric acid, dopamine and glycine. The depolarization can also occur due to an iPSP if the reverse potential is between the resting threshold and the action potential threshold. Another way to look at inhibitory postsynaptic potential is that there are also a chloride conductance change in the neuronal cell because it decreases the driving force. This is because if the neurotransmitter released into the synaptic cleft, it causes an increase in the permeability of the postsynaptic membrane to chloride ions by binding to the ligand-gated chloride ion channels and therefore causing them to open. Then the chloride ions, which are in greater concentration in the synaptic cleft, will diffuse into the postsynaptic neuron. As these are negatively charged ions, hyperpolarization results, making it less likely for an action potential to be generated in the postsynaptic neuron. The microelectrodes can be used to measure this postsynaptic potentials at either excitatory or inhibitory synapses. In general, a postsynaptic potential is dependent on the type and combination of receptor channel, reverse potential of the postsynaptic potential, action potential, threshold voltage, ionic permeability of the ion channel, as well as the concentration of the ions in and out of the cell. This determines if it is excitatory or inhibitory. The iPSPs always want to keep the membrane potential more negative than the action potential threshold and can be seen as transient hyperpolarization. EPSPs and iPSPs compete with each other at numerous synapses of a neuron. This determines whether or not the action potential at the presynaptic terminal will regenerate at the postsynaptic membrane. Some common neurotransmitters involved in iPSPs are GABA and glycine. So, what is the action of GABA in the development of inhibitory postsynaptic potential? This iPSP is the electrical potential in the form of hyperpolarization that develops during postsynaptic inhibition. The inhibitory neurotransmitter substance acts on postsynaptic membrane by binding with the receptor. The transmitter-receptor complex opens the ligand-gated potassium channels instead of sodium channels. Now, the potassium ions, which are available in plenty in the cell body of postsynaptic neuron, move to the ACF, that is extracellular fluid. Simultaneously, chloride channels also open and the chloride ions move inside the cell body of the postsynaptic neuron. The exit of potassium ions and influx of chloride ions cause more negativity inside, leading to hyperpolarization. The hyperpolarized state of the synapse inhibits synaptic transmission. So, we are going to see about presynaptic or indirect inhibition. It occurs during the failure of presynaptic axon terminal to release sufficient quantity of excitatory neurotransmitter substance. It is also called indirect inhibition. The presynaptic inhibition is mediated by axo-axonal synapse prominent in spinal cord and regulates the propagation of information to higher centers in brain. Normally, during synaptic transmission, action potential reaching the presynaptic neuron produces the development of epsp in the postsynaptic neuron. But in spinal cord, a modulatory neuron called presynaptic inhibitory neuron forms an axonic synapse with a presynaptic neuron. This figure shows the presynaptic inhibition. The inhibitory neuron inhibits the presynaptic neuron and decreases the magnitude of action potential in presynaptic neuron. The smaller action potential reduces calcium influx. This in turn decreases the quantity of neurotransmitter released by presynaptic neuron. So, the magnitude of epsp in postsynaptic neuron is decreased, resulting in synaptic inhibition. Renshaw cell or negative feedback inhibition. It is caused by Renshaw cells in spinal cord. The Renshaw cells or smaller motor neurons present in anterior gray horn of spinal cord. Anterior nerve root consists of nerve fibers which leave the spinal cord. These nerve fibers arise from alpha motor neurons in anterior gray horn of the spinal cord and reach the effector organ muscles. Some of the fibers called collateral fibers terminate on Renshaw cells instead of leaving the spinal cord. When motor neurons send motor impulses, some of the impulses reach the Renshaw cell by passing through collaterals. Now, the Renshaw cell is stimulated. In turn, it sends inhibitory impulses to alpha motor neurons so that the discharge from motor neurons is reduced. A Renshaw cell may be supplied by more than one alpha motor neuron collateral and it may synapse on many motor neurons. This figure shows the Renshaw cell inhibition. In this way, Renshaw cell inhibition represents a negative feedback mechanism. A Renshaw cell may be supplied by more than one alpha motor neuron collateral and it may synapse on many motor neurons. Next, we are going to see about feed-forward inhibition. The feed-forward synaptic inhibition occurs in cerebellum and it controls the neuronal activity in cerebellum. During the process of neuronal activity in cerebellum, stellate cells and basket cells, which are activated by granule cells, inhibit the Purkinje cells, thereby releasing GABA. This type of inhibition is called feed-forward inhibition. And now about reciprocal inhibition. It is the inhibition of antagonistic muscles. When a group of muscles are activated is called reciprocal inhibition. It is because of the reciprocal innervations. Coming to the significance of synaptic inhibition. The synaptic inhibition in the central nervous system limits the number of impulses going to muscles and enables the muscles to act properly and appropriately. Thus, the inhibition helps to select exact number of impulses and to omit or block the excess ones. When a poison like strychnine is introduced into the body, it destroys the inhibitory function at the synaptic level, resulting in continuous and convulsive contraction even with slight stimulation. In the nervous disorders like Parkinsonism, the inhibitory system is impaired, resulting in rigidity. Coming to the properties of synapse. One-way conduction or Bell-Magendy's law. According to Bell-Magendy's law, the impulses are transmitted only in one direction in synapse, that is, from presynaptic neuron to postsynaptic neuron. Synaptic delay. It is the short delay that occurs during the transmission of impulses through the synapse. It is due to the time taken for release of neurotransmitter, passage of neurotransmitter from axon terminal to postsynaptic membrane, action of the neuron transmitter to open the ionic channels in the postsynaptic membrane. Normal duration of synaptic delay is 0.3 to 0.5 millisecond. The synaptic delay is one of the causes for reaction time of reflex activity. Significance of determining the synaptic delay helps to find out whether the pathway for a reflex is monosynaptic or polysynaptic. Fatigue. During continuous muscular activity, synapse becomes the seat of fatigue along with bed cells present in the motor area of frontal lobe of cerebral cortex. Fatigue at the synapse is due to the depletion of neurotransmitter substance acetylcholine. The depletion of acetylcholine occurs because of the two factors, that is, 1. Soon after the action, acetylcholine is destroyed by acetylcholine esterase, and due to the continuous action, new acetylcholine is not synthesized. Summation. It is the fusion of effects or progressive increase in the excitatory postsynaptic potential in postsynaptic neuron when many presynaptic excitatory terminals are stimulated simultaneously or when single presynaptic terminal is stimulated repeatedly. Increased epsp triggers the axon potential in the initial segment of the axon of the postsynaptic neuron. Summation is of two types, spatial and temporal. Spatial summation occurs when many presynaptic terminals are stimulated simultaneously, whereas temporal summation occurs when one presynaptic terminal is stimulated repeatedly. Thus, both spatial summation and temporal summation plays an important role in the facilitation of response. Electrical properties of the synapse are the epsp and ipsp, and convergence and divergence. Convergence is the process by which many presynaptic neurons terminate on a single postsynaptic neuron, and divergence, it is the process by which one presynaptic neuron terminates on many postsynaptic neurons. To conclude, nervous system as a series of nerve impulses which transmits information. The movement of an action potential as a wave through a nerve fiber to nerve impulse. A wave of a negative charge on the surface of an axon marks the position at any moment of the action potential. Action potentials are increased as well as self-generated along the axons. The close proximity of the axon of one neuron and the dendrite of another neuron with a gap of just about 200 in between the synapse. Structure of the neuroglandular junction and the transmission of nerve impulse across them are similar to those of neuron to neuron synapse. Thank you.